Good morning, everybody. Welcome to this edition of Grand Rounds. Thank you for joining us this morning. It is my great pleasure to introduce Dr. Kristy Brown, who is joining us from Weill Cornell Medicine in New York. Dr. Brown received her PhD in Montreal, where she studied reproduction, and then headed to Australia, where she spent some time there gaining expertise in the areas of steroid hormone receptors, metabolism, breast cancer, obesity, and then started her faculty position at Weill Cornell in 2017 and is now an associate professor of biochemistry and medicine there. She has published some really nice, beautiful work on sort of defining these very complex mechanisms and that occur that sort of help define this link between obesity and breast cancer, and we'll hear about that today. This is actually the first time I've met Dr. Brown. I think Grand Rounds is a great opportunity to bring in new people and hear about explore new areas, and sometimes it's a great way to build collaborations. I had actually run across her work. She has a really nice website, actually, that's very up to date, which I'm kind of jealous of. We need to work out ours. She's based on some of the directions that my lab's going in, I sort of ran across her website and her work, and it turned out that she knew some people, some of the breast cancer researchers here. So I thought it'd be great to have her out and give a talk. And we look forward to seeing your talk and meeting with you later today. Thanks so much for joining us this morning.
Thank you so much, and thanks for the invitation. I was really pleased to receive this because I feel like, we don't always have an opportunity to present this work broadly, and I think in the Zoom era we're able to do that a lot more. So as Kaylee was mentioning, I'm interested in the link between obesity and breast cancer, and for a long time we were really focusing on the mechanistic link. But what that has taught us is that we can use those findings really to start thinking about new strategies for intervention. So today what I thought I would do is give you a bit of background in relation to the mechanistic findings that led to the study of a gut-derived hormone, which we're now considering using in the clinic. So let's see. So this will come as no surprise to most of you in the field, especially that breast cancer is a very important health concern, not only in the US, but globally. And internationally, it affects 2.1 million women each year with new diagnoses. And it's considered the most frequent cancer among women, especially in developed countries. Clinical decision-making is largely based on the biomarker expression of 3 receptors, the estrogen receptor and the progesterone receptor, as well as HER2. And the reason is that we can actually act on these biomarkers. So we know that ER and PR expression are predictive of potential response to endocrine therapy, and HER2 expression can be used to predict whether someone will respond to Herceptin, an anti-HER2 therapy. At the moment, all other tumors are classified as triple-negative breast cancers, and you can imagine that, in fact, they're much more heterogeneous than this, but these cancers don't have currently approved targeted therapies. One exception to that is PARP inhibitors that are used for the treatment of BRCA mutant tumors or in BRCA mutation carriers. And these are showing quite a bit of effectiveness in this population. There is still a strong need to identify therapies for this population, though. That being said, the majority of breast cancers do express the estrogen receptor, and endocrine therapy involving aromatase inhibitors, selective estrogen receptor modulators or degraders are first-line therapy for women with ER-positive breast cancer. At our center, as well as many others, these are now offered in combination with CDK4/6 inhibitors. These inhibitors are used because they're one of the main mechanisms of resistance to endocrine therapy. Other mechanisms of resistance include mutations in the estrogen receptor itself. And the use of these this therapy, even though it's relatively effective, is also associated with side effects that impact a large proportion of women who take the treatment. So it's not a given that someone who's being treated for breast cancer, even though they will likely do well on the therapy, that they will have a reasonable quality of life. And arthralgia or joint pain is the #1 complaint amongst women who take endocrine therapy. And this leads to about 1/3 of these women to discontinue the use of these therapies. And obviously that's associated with an increased risk of recurrence and potentially death. So there is still a need to be looking at alternate therapies, safer and effective treatments for all of these breast cancers. And that's one of the areas that we've become interested in over the years. So today I'll be talking about a number of different aspects that, or findings that have come out of our laboratory. I'll talk mainly about the gut-derived hormone Ghrelin and its unacylated form, unacylated Ghrelin, which we found suppress inflammation and aromatase expression in the breast microenvironment. So they do have an effect in the microenvironment that can lead to suppressing the growth of cancers. We've also found that unacylated Ghrelin suppresses the growth of many breast cancer subtypes, including not only hormone receptor positive breast cancers, but also HER2 positive and triple negative breast cancer, as well as endocrine therapy resistant breast tumors. What was interesting from these studies, and I'll highlight throughout my talk, is that the effects of unacylated Ghrelin were dependent on us culturing cells in a 3D matrix that was biologically relevant. So at the time that we started these studies, it wasn't obvious that the ECM was important as far as mediating response to treatment. But this is something that we discovered whereby these peptide hormones had effects in 3D, but not in 2D. And it really led us to think about the implications of this study as well as other similar studies for cancer biology and drug discovery. We're often, maybe less so now, but in the past we were mainly relying on 2D cultures of breast cancer cells to predict response to treatment. So I'm going to take a major step back here and talk a little bit about estrogens, because a lot of the work that we do is really aimed at identifying safer treatments for breast cancer. And the reason for this is that endocrine therapy that targets either estrogen production or estrogen action inhibits the effects of estradiol in particular, not only in the breast, but throughout the body. And this has a number of consequences, that not only impact the quality of life of women, but can also lead to comorbidities that impact the lifespan of women. So this here, I don't know if you can see my pointer, but I'll assume that you can. On the top left here, we have the chemical structure of estradiol. Estradiol is the most active estrogen that our body produces. It binds with the highest affinity to the estrogen receptor. And, during reproductive years, it's predominantly produced by the ovaries in developing follicles and in ovulatory follicles. And with every cycle, we see an increase in estrogen levels and a reduction. And this cyclic nature of estradiol production appears to be important for the normal functioning of many organs in the body, not only for women, but also, estrogens in men are also very important. So estrogens are recognized, and most of us will appreciate their effects in the reproductive system, where they're involved in stimulating breast development during puberty, but also increasing receptivity of an embryo at the time of, in the uterus, so preparing the uterus for implantation. And obviously have an effect in the breast to stimulate proliferation after breast development as well. Beyond effects on reproductive tissues, we know that estrogens are important to preserve bone density. They're important for the cardiovascular system and and the liver, and they've also been implicated in cognitive function in the brain. So as you can imagine, if we stop the synthesis or the action of estrogen, we will be impacting a large number of these organs. So this, I like to use this image because it reminds me of how the mammary gland behaves over a woman's life cycle, if you will. And I think Kaylee is probably very well placed to talk about, breast development and regression. But I like to show this because it really reflects the changes that the mammary gland will undergo, like I said, during a woman's lifespan. So at puberty, we do see more development of the mammary gland that is receptive to the effects of hormones during pregnancy to really cause differentiation of the mammary gland in preparation for lactation. And at the time of weaning, this mammary gland actually will regress. So many of the cells will undergo apoptosis, and there'll be a very dramatic remodeling of the breast, not only the breast gland, but also the microenvironment of the breast. And to a lesser extent, this also happens with every menstrual cycle with changes in hormones, where we do see changes within the gland. At the time of menopause, however, the gland actually does regress substantially and is thought to be quiescent. So it's quite interesting to think that the majority of breast cancers actually occur at this time when the gland itself is quiescent. So this is largely due, to a plummeting of hormone levels after or during the menopausal transition. And this change in hormone levels is also associated with other effects throughout the body. So at the time of menopause, estrogen levels decrease, but androgen levels actually stay relatively stable. And as a consequence, the ratio of these hormones changes during the menopausal transition. And this is also associated with changes in metabolic aspects, where a woman who is previously pear-shaped and accumulated fat around the waist, around the hips and the thighs, will all of a sudden accumulate weight around, centrally around the waist, leading to more of an apple shape. This is obviously associated with worse metabolic outcomes, increased risk of diabetes, as well as metabolic syndrome. And unfortunately, obesity after menopause is also recognized as a risk factor for hormone receptor positive breast cancer. So this is just an image from Cancer Research UK showing that, we now have strong evidence linking obesity to the development of 13 types of cancer. And this includes, hormone receptor positive breast cancer after menopause. So it's somewhat counterintuitive that the majority of breast cancers have to happen after menopause, and that these are hormone receptor positive when we know that circulating estrogen levels decrease at that time. One way to kind of look at this is to have a better understanding of the breast itself, because it's hypothesized that many of the changes that occur within the breast at the time of menopause may be contributing to the development and the growth of breast cancers after menopause. So this is a sagittal section of the female breast, and as you can see, we clearly see these tree-like structures, which are the breast ducts and the site of origin of breast cancers. And these ducts will interact quite a bit with its surrounding adipose tissue. The adipose, is substantial and in almost direct contact with these glands, and is hypothesized to, really drive the development and the growth of tumors within the breast. So our lab and others have been very much interested in how the adipose tissue changes over time and in relation to menopause, but also in the presence of breast tumors. And we focused a lot of work on trying to understand how the aromatase enzyme is regulated during the menopausal transition, but also in the context of breast cancer. Aromatase catalyzes one of the last steps in estrogen biosynthesis by converting androgens like androstenedione and testosterone to estrogens like estrone and estradiol. And interestingly enough, at a time when circulating levels of estrogens are low, the breast adipose actually is able to produce estrogens from the conversion of androgens via the effects of aromatase within the breast tissue. So after menopause, not only does the adipose tissue do this, but the other extragonadal sites, such as the bone and the brain, can also support the normal functioning of tissues, which is why, it's important to be able to maintain the function of these tissues and the local expression of estrogen to be able to prevent, deterioration of these tissues. This is an example of aromatase staining in brown in a breast tumor sample, where we see here is a ductal carcinoma in situ. So tightly packed tumor cells adjacent to adipose tissue. Here we see the lipid-laden adipocytes. And we also notice that aromatase is highest in these cells that are immediately adjacent to the tumor. It turns out that most of these cells are actually adipose stromal cells, or pre-adipocytes, so precursor cells to these mature adipocytes. And what happens is, this is an example of desmoplasmic reaction, where the cells are really accumulating adjacent to a tumor. And this is a consequence of the increased proliferation of these adipocyte precursors. It's also hypothesized that many of the factors that are produced by tumors can prevent the differentiation of these cells into mature adipocytes, and can potentially even cause the de-differentiation of the mature fat cells into these adipocyte precursors, or adipose stromal cells. And interestingly, these are the sites of highest expression of aromatase, especially in the context of breast cancer. So the increased expression of aromatase here is hypothesized to occur as a result of response to factors that are produced by the tumor itself. And as a consequence, we get this feed-forward loop between the tumor and the stroma, leading to an increase in estrogen production that can then feed the tumor once more. Our lab has developed a number of immunofluorescent methods to be able to semi-quantitatively assess the staining for aromatase. It can be a bit tricky in the sense that we're really wanting, to be able to characterize staining within different cells. And this is work that we initiated a long time ago. I guess we started characterizing stromal specific expression about 10 years ago now, and we developed algorithms to be able to select adipose stromal cells specifically based on the shape of the cells, etc. And by doing that, what we found was that aromatase immunoreactivity, and here we see it in red, and it might be a bit hard to see, but the stromal cells are the elongated fibroblast-like cells, and aromatase is expressed in the endoplasmic reticulum, so the red staining that surrounds the nucleus here. Here we have the blue nuclei in the inset. And by quantitating this signal, we were able to demonstrate that aromatase levels on a per cell basis are positively correlated with the body mass index of women. So we were showing that, with increased obesity, we were seeing higher levels of aromatase in the cells, suggesting a regulation of expression within these cells. So beyond having more of these cells around, we were also seeing that with obesity, there was more aromatase within these cells themselves. So we and others have spent a lot of time trying to characterize the mechanisms of aromatase regulation within the stroma and how this contributes to tumor development, but also more broadly, how the adipose tissue interacts with the tumor cells to be able to stimulate tumor development and growth. And this is just a cartoon diagram highlighting some of these relationships. We didn't include the CM in this particular image, But we do see that the adipocytes, which are responsible for producing adipokines, including leptin and adiponectin, are able to contribute to the growth of these tumors. We know that inflammatory mediators that are produced by immune cells, in particular macrophages, can drive the expression of aromatase, as can these adipokines, and in response can then act on the epithelium to drive the growth of tumors. So in studying aromatase regulation within the fat, we obviously needed to have some understanding of the aromatase gene. And what makes it interesting is also what allows us, to potentially start thinking about targeting aromatase in a breast-specific way to try to eliminate some of these side effects that we're seeing. So the reason I'm saying this is that the aromatase gene, or the CYP19A1 gene, is relatively complex. It's composed of 9 coding exons, which are the same throughout the body, but it is also composed of promoter selective or tissue selective first exons, which are driven by tissue specific promoters. So what happens is that in the breast, we have a coordinated activation of promoters 1.3 and 2, leading to the increased expression of aromatase in both obesity and breast cancer. Whereas in bone, for example, it's promoter 1.4 that's used to drive expression. So the fact that this gene has this level of complexity allows us to start thinking about, transcriptional regulators of the gene as potential novel inhibitors, as opposed to inhibiting the catalytic activity of the enzyme. Now, through the years, we've identified many factors, as I mentioned in the previous slide, that can alter the expression of the aromatase gene, including adipokines produced by the adipose tissue, inflammatory mediators, and more recently, we also became interested in hunger-regulating hormones, and this is a lot of the work that I'll be talking about today. So without going into too much detail about the biochemistry here, because all of this is published, I thought I would present you a slight overview of the metabolic pathways that we've examined over the years for their role in regulating aromatase expression within the breast. So we identified relatively early on that AMP kinase, which is an energy sensor in most cells, as it can directly bind to ATP or AMP levels, and alter pathways of energy utilization and production. We found that AMP kinase was actually a key negative regulator of aromatase within the breast. I'm going to spare you the details of how we got to this, but it was really based on hereditary cancer syndrome studies, etc., which you can read more about in our publications. But what we found was that AMP kinase was able to suppress aromatase by acting on Krebs signaling. So it inhibited the nuclear localization of this Krebs co-regulator, and Krebs is required for the activity of promoters 1.3 and 2, those breast selective promoters. Over the years, we've extended these studies to include P53 as a negative regulator of aromatase as well, whereas HIF1 alpha and PKM2 were found to be drivers of aromatase expression within the breast. So characterizing these pathways, which are also known to be important in cell metabolism, provided us a new link between obesity and breast cancer via the regulation or the dysregulation of these metabolic pathways in the microenvironment itself. And at the time, the microenvironment metabolism hadn't really been studied extensively. So we were really excited to be able to make this contribution at the time. What we found was that many of the factors that are also altered in obesity, including prostaglandin E2, adiponectin, and leptin, acted on these pathways to regulate aromatase expression, with prostaglandin E2 and leptin being elevated in obesity and driving aromatase, and adiponectin being able to counter the effects of these of these factors. And this was also an indication to us that we could be targeting this pathway to potentially inhibit aromatase expression within the breast. With regards to appetite regulating hormones, we had studied leptin relatively, extensively previously, and then we became interested in another hormone that had opposite effects on appetite. So the leptin ghrelin system is really important as far as appetite regulation, with leptin being a satiety hormone. So with a full stomach, or after eating, leptin levels are higher, and this signals to the central nervous system to stop eating. On an empty stomach, however, our stomach itself will produce a hormone called ghrelin, and this hormone will stimulate appetite until the stomach is distended and ghrelin levels are no longer produced, or ghrelin is no longer produced. So this balance between ghrelin and leptin is really important as far as regulating how we normally consume food. But this is really only true in healthy individuals. So in people who do not have, issues of metabolic syndrome, or are overweight or obese. With increasing BMI, what we do see is that we develop leptin resistance, and as a consequence, leptin can no longer stop appetite. And as a consequence, we get this feedback mechanism where leptin, which is produced by the fat cells, actually increases with BMI. And even though the central nervous system is resistant to the effects of leptin, the rest of our bodies is not. When it comes to ghrelin, however, things are slightly different in the sense that this appetite stimulating hormone is decreased in circulation with increasing BMI. And the method of regulation there is pretty complex. It has a lot to do with how ghrelin levels change before and after feeding as to whether it will stimulate appetite or not. And I'm not going to go into that today. But what was interesting to us was that this hormone that is known to stimulate appetite by stimulating AMP kinase in the hypothalamus is actually decreased with BMI. So because of that link with both AMP kinase regulation and obesity, we became interested in seeing whether this hunger hormone may also be involved in regulating estrogen production within the breast. So before I go into those studies, I thought I would tell you a little bit more about ghrelin and its unacylated form. So ghrelin and unacylated ghrelin are 28 amino acid peptide hormones. Ghrelin is the best characterized of the two. It has this primary amino acid structure here and has an octanoyl group on its third serine. And this is the peptide hormone that binds to the cognate ghrelin receptor, GHSR1A, in the hypothalamus to regulate appetite. Both of these hormones are produced by a prepro hormone called preproghrelin, which is here. And depending on how much of this hormone is produced, an enzyme called GOAT will actually acylate or add this octanoyl group on the prepro hormone, which can then be further cleaved into the 28 amino acid peptide hormone as acylated ghrelin or ghrelin, as well as unacylated ghrelin, so the unacylated form. So as I mentioned, the acylated form of ghrelin will bind to its cognate receptor in the hypothalamus, GHSR1A, to regulate appetite. And for the longest time, people thought that unacylated ghrelin was a byproduct of this process and didn't really have any biological effects. But more recently, it was described that unacylated ghrelin has a number of beneficial effects metabolically. It does not bind to the cognate ghrelin receptor, but is hypothesized to bind at a yet unidentified receptor. And both of these peptide hormones have different effects physiologically. So here you see some of the effects of ghrelin. Ghrelin actually stimulates insulin resistance. So as you can imagine, ghrelin is produced at times of low energy or low food availability. So really it's a conservation method. But unacylated ghrelin tends to increase insulin sensitivity and decrease postprandial glucose levels in both humans and rodents. So they have very, very different effects. And unacylated ghrelin appears to be more beneficial if we're thinking about, someone who may have insulin resistance. So unacylated ghrelin can counter those effects. So getting back to our hypothesis, we thought that ghrelin, because it acted via GHSR1A in the hypothalamus, to activate AMP kinase, and the result was, stimulation of growth hormone release and increased appetite, that ghrelin may in fact be able to activate AMP kinase in the breast microenvironment and suppress aromatase expression. At the time, we knew that unacylated ghrelin didn't do that in the brain, so we thought we would use it as a negative control. So when we tested the effects of ghrelin in isolated adipose stromal cells, and these are all human primary cells because the mouse and the human are quite different when it comes to aromatase regulation. So we tend to favor using human cells whenever we can. But here you see a dose-dependent decrease in aromatase expression as was expected initially in response to increasing doses of ghrelin. And here we see we're dealing with 10, 100, and 1,000 picomolar of ghrelin. So this is relatively low, but it is within the physiological range of ghrelin levels, which is, in the hundreds of picomolar. And as you can imagine, with obesity, we actually see a reduction in these levels. So this was giving us a sense that, with obesity, where we have lower levels of ghrelin, it may not be able to suppress levels of aromatase so effectively, and this may contribute to our understanding of why aromatase levels are higher in obesity. What was also interesting was that ghrelin was able to counter the effects of one of the main stimulators of aromatase in the breast, prostaglandin E2. So here we see that prostaglandin E2 can stimulate aromatase expression within these adipose stromal cells, and that 10 picomolar of ghrelin is able to suppress that. And this was getting us to think about, well, could we actually be using this clinically to suppress aromatase in the breast? At the time, however, we weren't exactly sure how it was working. In the hypothalamus, we knew that ghrelin acted on GHSR1A to activate G-alpha-Q coupled GPCR, leading to an increase in intracellular calcium. But we weren't seeing that in our stromal cells. So we started looking at alternate second messenger systems. And interestingly enough, we found that ghrelin was able to suppress both the forskolin and the basal levels of cyclic AMP within the cells. And cyclic AMP is a major regulator of Krebs, obviously. So this was providing us a link between, ghrelin, cyclic AMP, Krebs, and aromatase regulation within the breast. So this is just to remind you that promoters 1.3 and 2 are really dependent on the effects of cyclic AMP, whereas other promoters are not. So this would suggest that ghrelin would act specifically on the breast promoter of aromatase. Where things became a little bit more complicated was that what was supposed to be our negative control, decyl ghrelin or unacylated ghrelin, which we didn't expect to have an effect because it was known not to bind to the cognate receptor, was also able to suppress aromatase expression within the adipose stroma. And this led us to start thinking about these alternate ghrelin receptors that had previously been proposed as being important for binding both unacylated ghrelin and ghrelin. So can we potentially be using, decyl ghrelin or decyl ghrelin analogs to better treat estrogen-dependent breast cancer? Well, following on from what I just mentioned, we were seeing effects of these peptide hormones on cyclic AMP and the regulation of aromatase within the adipose stromal cells. We also wanted to see whether there were effects on the osteoblasts or the cells of the cartilage that are responsible for expressing aromatase. what we found was that there was no significant effect of unacylated ghrelin on aromatase within the osteoblast, suggesting that these peptide hormones would not suppress estrogen production at other sites, other than the breast. In order to start thinking about, treatment, we also wanted to look at what was happening within breast tumor samples. And here is, again, focusing on aromatase activity. These are breast cancer explants, if you will. So these are pieces of breast tumors that include not only the tumor cells, but also the adipose stroma. In this case, they're about, I think we were dealing with like a 0.3 to 0.5 centimeter cube piece of tissue that was cultured on top of surgical sponges. This is a method that was adapted in Wayne Tilley's lab in Adelaide to allow the tissue to interact with the media via the sponge as opposed to being submerged and risk, increased hypoxia of the tissue. So we adapted this method to be able to assess aromatase activity using the tritiated water release assay. So if we just look at the explants, what we found was that breast cancer explants have much higher aromatase activity than cancer-free explants. This was not a surprise to us. We knew that tumor-derived factors were able to drive aromatase expression in the stroma, and that tumor-bearing tissue actually had higher estrogen production. We used letrozole as a positive control. Letrozole has an IC50 of 11 nanomolar. So here we tested 1 at 10, and we were limited on the amount of tissue for these studies. So we had to limit the number of doses we were testing. But letrozole, as expected, had a dose-dependent effect on aromatase activity within these explants. What was exciting to us was that desicloghrelin or unacylated ghrelin also caused a dose-dependent decrease in aromatase activity in these breast cancer explants, but at 10 and 100 picomolar, so 100 times lower doses than letrozole. So this was encouraging for us to start thinking about unacylated ghrelin, which has all these beneficial effects on metabolism, has very few side effects in the studies that had been done previously to look at effects on diabetes, for example, was able to reduce aromatase expression in stromal cells in isolation, but was also able to suppress aromatase activity in breast cancer explants, to start thinking about how we could actually leverage this finding for better treatments in the clinic. So before doing that, we really wanted to start thinking about, what desicloghrelin may also be doing to the cancer cells themselves. And here we're switching gears a little bit. So up until now, we were really focusing on aromatase regulation, aromatase activity. But for the next part of my talk, the findings are really in relation to tumor cell biology. So this is independent of effects on aromatase expression, because we wanted to see whether unacylated ghrelin also had independent effects on the tumor cells that may either contribute or counteract the effects on aromatase expression. So there had been a couple of studies looking at the effects of ghrelin and unacylated ghrelin on breast cancer cell proliferation, but not much really. At the time, this study is really looking at a number of different ghrelin and ghrelin analogs on, this was thymidine incorporation, so looking directly at proliferation of MCF7 cells. And we see here with ghrelin, which is, these black boxes, that there is a dose-dependent decrease in cell proliferation, in MCF7 cell proliferation. But, we're really needing, close to micromolar doses to achieve a 50% reduction in growth. And if we think about desicloghrelin or unacylated ghrelin, which we think is probably more beneficial from the point of view of treatment, the effects were even less pronounced. That being said, before thinking about, clinical use of desicloghrelin or unacylated ghrelin, we really wanted to revisit these studies in our own lab to get a better idea of, what the impact would be of picomolar doses, which according to this study would have no effect. So interestingly enough, the student who was responsible for this study at the time treated our cells in 2D at the time, and came to me and said, well, I'm a bit worried about the findings from this study, because it seems that that unacylated ghrelin is really affecting the adherence of the cells to the plastic. And I feel like this might be skewing the results in one way or another. So with every wash, there were differences between, the positive control, the negative control, and unacylated ghrelin as to how the cells were behaving. And we thought, well, let's have a closer look. We really want to see what's happening to the full population, whether we can detect apoptosis or anything else. So let's grow the cells in Matrigel. And at the time, this was, a combination of, we want to keep the cells there, and if they're grown in Matrigel, they can't really float away. But it was also a time when people were becoming more interested in using extracellular matrix in trying to understand tumor cell biology and treatment. So we thought, well, this is a good time for us to start exploring that. So I encourage the students to study the cells in 3D as opposed to 2D. And what was interesting was that, so this is the effect of just looking at cell number, how cells grow in the presence of serum, which is not unexpected. This is in 2D. And this is the effect of 100 picomolar of unacylated ghrelin. We were seeing, some reduction, but nothing dramatic here. As soon as the cells were grown in 3D, the cells tended to grow or to proliferate faster. And the suppression with unacylated ghrelin was very dramatic. And, we got really, really excited when we first saw this finding. Obviously, we had a lot more work to do to try to figure out what was going on. But this was the basis for the paper that we published last year in this field. So we basically compared, the effects of unacylated ghrelin to chemotherapeutic agent doxorubicin in ER positive as well as triple negative cell lines. We see the doxorubicin causes a dose-dependent decrease in cell number. This is in 3D. In both cases, so the cells are responsive to doxorubicin at relatively low doses. Unacylated ghrelin, on the other hand, had much more potent effects, but unacylated ghrelin only suppressed the serum stimulated growth. So these two dotted lines are the range of cell proliferation of serum containing wells versus serum starved wells. So really what we were seeing was a suppression of cell proliferation, but no induction of apoptosis. At least this is what we were inferring from these studies here. We were curious to see how broad the effects were of unacylated ghrelin, and this is a panel of breast cancer cell lines, including luminal A, so ER/PR positive, luminal B, which also express HER2, and HER2 positive cells. The black bars reflect the growth of cells in the presence of serum, and so without and with 100 picomolar of unacylated ghrelin. And all of these cell lines responded to the effects of unacylated ghrelin. Here we have LCC2 cells, which are a derivative of MCF7 cells that are resistant to tamoxifen. So all of a sudden we were introducing this concept that endocrine therapy resistant cells were also sensitive to the effects of unacylated ghrelin. When we looked at triple negative breast cancers, however, the effects were a little bit more varied. Again, we have a number of different triple negative cancers of different, subtypes, either basal or mesenchymal. And some of these cells responded to the effects of unacylated ghrelin, but others did not. So here we have 2 out of the 5 that responded. The non-responders ended up having, we discovered that they had mutations in BRAF, HRAS, and KRAS, which is part of the MAP kinase signaling pathway. And we thought that this might be a mechanism of resistance, and it may in fact be downstream of unacylated ghrelin signaling. So we were really interested in trying to figure out how unacylated ghrelin was acting. Now, keeping in mind that we don't know what the receptor for unacylated ghrelin is, we hypothesized that it was similar to what we were seeing in the stromal cells, and it might be a G-alpha I coupled GPCR, because in those cells it suppressed cyclic AMP levels. But beyond that, we know very little about how unacylated ghrelin works. We also looked at primary tumors. This is one example of an ER-positive primary tumor. We looked at 2 of them, and both were responsive to the effects of unacylated ghrelin. And in triple-negative breast cancers, again, 2 of our cancers were responsive and 2 were not. We didn't actually have mutation status for these patient-derived cancer cells, but we did have microarray analysis of RNA expression for these 4. And we were able to define a signature of response to unacylated ghrelin. So this can be useful for a couple of different reasons. First of all, it seemed to be that the non-responders had activated MAP kinase signaling, which may be consistent with a mutation in BRAF or KRAS. The other thing is that we could actually start thinking about using this signature with patient samples to be able to, identify populations that may be responsive clinically. So because of the consistent findings of, potential role of MAP kinase signaling in these studies, we decided that we really wanted to characterize the signaling a little bit better here. Keeping in mind that all of our studies needed to be done in 3D. So for anyone working in cell culture, when we're using Matrigel, it's not so obvious to translate all of our Western blotting findings. We've also done CRISPR library screens with this. It took a bit of time to optimize these studies, but in the end, I think we got there. This is an example of how to study, or potentially how to study, MAP kinase signaling and AKT signaling in 3D. So we obtained an ERK reporter system. So this is a FRET-based system where we can transfect our cells beforehand and then culture them in 3D. And the FRET-based system is really interesting in the sense that if ERK is active, it will phosphorylate the substrate, leading to a folding of the protein on itself. And this actually brings both of our fluorophores together. And as a result, you can actually look on a per cell basis what the activation of ERK is within the cells. So we use this construct in our cells and looked at the effects of unacylated ghrelin, as well as a MEK inhibitor, UO126. And what we found was that, over time, serum does stimulate ERK activity as expected, but this is suppressed in the presence of the MEK inhibitor as well as unacylated ghrelin. So really unacylated ghrelin was able to suppress MAP kinase signaling. And this might be, the mechanism of action of unacylated ghrelin. Here, looking at a more traditional Western blot, again, from cells isolated in 3D culture, we do see a suppression of phosphorylated ERK at 5 and 15 minutes, as well as ERK target, phospho90, P90RSK. But complementary to this, we were also seeing effects on AKT signaling. And the one thing to remember is that the signaling in 3D is actually slightly different to what we know in 2D, in the sense that in 2D cultures, it seems that MAP kinase signaling and PI3 kinase AKT signaling are kind of like in a yin-yang situation where if one of them is turned on, the other one is usually turned off. But when we look at cells in 3D, both pathways seem to be regulated concurrently. So here we see activation of both MAP kinase and AKT signaling in 3D. And both of these are suppressed by unacylated ghrelin. So in order to start pushing this a little bit further, we decided to revisit the cyclic AMP findings to really make sure whether, if we were dealing with G-alpha-I coupled receptor. Consistent with the stromal cells, we were seeing a reduction in cyclic AMP. We were also seeing activation of G-alpha-I. This is a pull-down experiment looking at G-alpha-I bound to GTP, which occurs with activation. And through a number of other studies using pharmacological as well as genetic approaches, we were able to characterize a signaling pathway that we think, maybe we think is representative of the effects of unacylated ghrelin. Of course, we still don't know what this receptor is, and we're still chasing that up at the moment. We also did in vivo studies. Here we see the effects in luminal A and luminal B xenograft. So these are in immunocompromised mice where the tumors, will grow in the absence of treatment, but with relatively low doses of unacylated ghrelin, we're able to suppress the growth of tumors. The J110 are a syngeneic model. These are in FVB mice. They're much more aggressive, and even though they're ER positive, in our hands at least, they're tamoxifen resistant. So, but in this case, we're also seeing effects of unacylated ghrelin. So in order to start thinking about this clinically, we have to think a little bit more about the practicality of using a peptide hormone. Unfortunately, peptide hormones are challenging in the sense that they have a relatively short half-life. They usually have to be administered intravenously, although that's not always the case. Fortunately, there was a company that was interested in developing unacylated ghrelin analogs for the treatment of Prader-Willi syndrome, which is a syndrome of overeating. And they were able to develop AZP531, which is a cyclic form of unacylated ghrelin. This form is much more stable than the linear form of unacylated ghrelin. It can be used subcutaneously, so not ideal when we're comparing to oral administration, but much better than an intravenous situation. And the drug was found to be extremely safe. So, very nice pharmacodynamic profiles and safety profiles. Unfortunately, the drug itself failed in clinical trials for Prader-Willi syndrome, but they were quite effective in a diabetes population, and we're trying to figure out whether we might be able to still use this for the treatment of breast cancer. So this is what AZP is. So it has, components that are common to both ghrelin and unacylated ghrelin. And what we found is that it's able to suppress the growth of breast cancer cells that we had found were sensitive to unacylated ghrelin, but is not effective in tumor cells that are actually resistant to unacylated ghrelin. So we know that the mechanism of action is likely similar between these two. It seems to be very effective at reducing the growth of primary breast tumors. Here we see the effects of AZP531 versus doxorubicin. And in vivo, it also has effectiveness in triple negative cancer as well as our syngeneic model. So we think that, based on these studies, as well as others, that we should be thinking more broadly when it comes to breast cancer treatment. We should not only be thinking about targeting the breast cancer cells themselves, but if we can combine that with the treatment of the microenvironment and potentially even the whole body metabolism. So if we're thinking about, obesity and metabolic syndrome, obviously that's associated with hyperinsulinemia, hyperglycemia that also contribute to the growth of cancers. So if we have a drug that potentially targets all of these aspects, we might be better placed to be able to tackle this issue. And it seems like many of the drugs that are targeting metabolism, including metformin, for example, are relatively safe. So I think this is something that we can be looking at also in the prevention setting. So this is something that we're thinking about at the moment as well. So with that, I'd like to acknowledge a number of people who were involved in this study. Shario was really, the grad student who's now a postdoc who led most of the unacylated ghrelin studies out of my lab. So she deserves a lot of credit here. I'd like to also acknowledge, the surgeons and the clinicians who were involved in allowing us to obtain many of the samples, including pathology. And, we don't say this enough, and I'm not saying this because this is a pathology crowd, but I think, for basic scientists to be able to access clinical specimens requires a large team effort, and I think that needs to be acknowledged. Our funding as well, obviously we've been fortunate to be able to undertake these studies, and I also want to acknowledge the women who donate their tissue for these studies. Without them, it would not be possible. Thank you very much.
Thank you, Kristy. That was a really beautiful story. I'm going to open it up. We have a couple minutes for questions. If anyone has any questions, you can unmute and ask.
Hi.
Hi, you and I are talking a little bit later today. We'll get in a little more depth, but one of the questions I had in your animal xenograft studies, does the unacylated ghrelin have any physiologic effects on the animals? Do they stop eating? Do they lose weight? So part of the question is, even though you don't know the receptor, does the UAG you use to actually interact with whatever the putative mouse receptor is?
Yeah, well, we don't know yet. So actually what's interesting here is that we use the rat unacylated ghrelin for our studies. We did compare human and rat, and they seem to be similar, but because we had historically done more studies with rat, we continued with the rat unacylated ghrelin. So that's the ghrelin that we're using here. To answer your first question, we saw no effects on the mice, no effect on mouse weight. We had pathology look at, the tissues, do a gross assessment of the overall, health of the mice. There was nothing that came up out of this. We have not done the studies though in, a model of diet-induced obesity or anything like that. And I think that's really where we should be pushing this, because I think the benefits might be even greater in those models.
Yeah, and I'll remind people of a very old literature, maybe only Leo knows this, but in the bad old days of endocrine-resistant breast cancer, I'm a medical oncologist, in the bad old days of endocrine-resistant breast cancer, we used to surgically ablate the endocrine system. So ovaries would come out first, and we, the University of Minnesota, contributed this literature. Then the adrenal glands would be removed, and then the last step was the pituitary. And actually, if you look at that old literature, removing the pituitary at that point, actually, you got some decent responses. I work on growth hormone and IGF, so I always say that's a growth hormone story, but maybe it's a Ghrelin story.
Yeah, and that's kind of why we stayed away from ghrelin, because we know that ghrelin stimulates IGF-1 secretion as well. So that was one of the reasons we stayed away from it. And I think that's also one of the reasons why it failed clinically as a treatment for cancer cachexia, is that, even though it can stimulate appetite, I don't know, I don't think anyone ever divulged the reasons why ghrelin stopped being used for cancer cachexia. But I always had a bad feeling about IGF-1 in this scenario, but unacylated ghrelin doesn't have that effect on IGF-1.
Okay, thanks.
Yeah, Leo.
Yeah, I have a question. So thanks for that absolutely excellent talk. And Doug was correct. I do remember those days. I also remember when cyclic AMP was everything 30 or 40 years ago. It was in every paper in Nature, whatever. So it was exciting. And cyclic GMP also. So a question for you. Do you know, or what are your suspicions about breast cancer in obese people versus obese people with diabetes? Is there any unique phenomenon that you've come across that you could make us aware of?
So I would, if I had to hazard a guess, I think, obese individuals with diabetes are are worse off, right? I think there's evidence for that already. We often study BMI, as a surrogate because we don't always have diabetes and insulin resistance information. But I think as far as these studies, and I think the metformin clinical trials are a good example of this, is that, I think the people who will do best on a metformin clinical trial are the individuals with diabetes or, some degree of insulin resistance. And I think the reason why a lot of these trials have been conclusive is because the populations were not selected for that reason in the long term. Also, the fact that they were that was given in addition to best practice, and as you know, that leads to very few cases to begin with of recurrence. So, I think the hyperinsulinemia effects, and obviously work from Dr. Cantley here at Weill Cornell highlights, the importance of PI3 kinase signaling in breast cancer. So I think it all works together. Yeah.
That's what I guess. Thank you.
All right. So I think unless there are any last minute questions, I think we have to wrap up and let Kristy get on with her day. And that was a fantastic talk. I think we look forward to meeting with you later today. And thanks for joining us this morning.
Thank you so much.